Hey everyone, welcome to Body Language. This is a video podcast that aims to dissect the intricacies of the human body. I'm Neha, and I've always been fascinated by the human body and its continual pursuit of keeping us alive. I'm by no means an expert, and I'm actually a really big fan of keeping things simple. So, with the help of doctors and specialists in the field, this podcast aims to break down the complexities that surround medical sciences into something more digestible for everyone. I hope you enjoy. What is aging? According to the Oxford Learner's Dictionary, aging is simply the process of growing old. On a surface level, aging may seem like this slow, inevitable process that all of us hopelessly aim to delay by employing various methods such as better skincare, exercise, and diets. Everyone always talks about how doing these things consistently will give you a longer, healthier life, but fewer people truly understand why. If someone runs every day, cuts out sugar from their diet, and generally maintains low levels of stress, how does this actually make them live longer? A potential answer to this question is the information theory of aging. Proposed by Dr. David Sinclair, the information theory of aging suggests that the process of aging is driven by ongoing loss of epigenetic information. Okay, so what is that? Before aiming to explain epigenetics, I need to recap the structure of DNA itself and also how it's stored in a cell. So, DNA. Simply put, it's a biological molecule that carries genetic information. DNA is something called a polymer, which just means that it's a chain that's made up of many small units that just repeat themselves. These units are called nucleotides. There are 4 different types of nucleotides, and genetic information that I just mentioned is determined by the order of the nucleotides in the DNA strand. This genetic information is further divided into smaller sections along the DNA strand, where each smaller section can code for the production of one specific protein. These smaller sections are called genes. When a cell needs to make a protein, it reads the sequence of nucleotides in a gene, and it uses that information to assemble the protein. So that's the structure of DNA itself. Now let's look at how it's actually organized in a cell. Each human body cell contains around 2 meters of DNA. Considering how small a cell is, it's really important that the DNA is stored very compactly so that all of it can physically fit inside the cell. For this reason, DNA strands coil themselves around proteins called histones. The coiled DNA then coils around itself again and again, and it eventually coils to form these condensed structures called chromosomes. A human diploid cell has 46 chromosomes, or sometimes people refer to it as 23 pairs of chromosomes. And all these chromosomes are located in the nucleus of the cell. This is how DNA is organized in a cell. Our bodies are comprised of many different types of cell. For example, neurons in the brain are different to keratinocytes in the skin, which are different to B cells in the blood. All these cells are completely different, but if you were to look into the nucleus of all these cells, you would find that they have the exact same genetic information, the exact same DNA sequence. This means that in a neuron or a brain cell, you would also find the genetic code to create a liver cell, or a white blood cell, or a muscle cell. So what I'm getting at is that every diploid cell in your body contains the genetic information to not just code for itself, but to code for every type of cell in your body. This then raises the question, how does a neuron know that it's supposed to become a neuron? All cells start as stem cells, and then they undergo a process called differentiation. If a stem cell is to turn into a neuron, it will only read and translate the genes for a neuron, despite having the genes for all cell types. By reading only these neuron genes, a stem cell differentiates into a neuron. The same principle applies to any other type of cell. If liver cells are to be formed, the stem cell reads and translates only the liver genes, and therefore it differentiates into a liver cell. But this doesn't really answer the question. What actually tells a stem cell you should only read the neural genes, or you should only read the liver genes? This is where epigenetics comes in. The selective reading of genes is regulated by epigenetics. Epi means above, and genetics refers to genes or DNA. So anything epigenetic involves modifications that sit above the actual DNA. These chemical modifications change gene activity without changing the actual DNA sequence itself. One of the main types of epigenetic modifications is called DNA methylation. DNA methylation involves adding something called a methyl group, which is basically a carbon covalently bonded to 3 hydrogen atoms, to certain parts of the DNA strand. This modification can turn genes on or off, helping to control which genes are expressed in a cell. So, going back to the concept of cell differentiation, DNA methylation indicates to the stem cell which genes to read and which to avoid. This tells the stem cell exactly what to do, so it can differentiate into a specialized cell. So, what does any of this have to do with aging? I previously said that aging is driven by the ongoing loss of epigenetic information. We now know that epigenetic information tells the cell precisely which genes to read and which genes to avoid in order to become a specialized cell. If this information is lost, then the cell does not know precisely which genes to read and which to avoid. This leads the cell potentially to read genes that it's not supposed to and avoid genes that it should be reading. This means that specialized cells slowly start to lose their identity, their specialty, and their function as they slowly start to read the wrong genes. This cellular miscommunication can result in cells becoming less efficient, malfunctioning, or even turning into senescent cells. Senescent cells are cells that no longer divide, and they accumulate in tissues, contributing to the overall decline in tissue function. Dr. David Sinclair coined the term X differentiation to describe this process of epigenetic information being lost. This is essentially the opposite of cell differentiation, which is when a cell becomes more specialized. This is because the process of X differentiation ultimately leads to the cell losing its specialization. When loads of cells altogether slowly start to lose their function, it manifests as visible signs of aging, such as wrinkles, gray hair, and loss of strength or muscle mass. On an even deeper level, it also results in the overall decline and health of tissues and organs, contributing to age-related diseases such as Alzheimer's, cardiovascular diseases, and even various forms of cancer. By viewing aging as a disease caused by X differentiation, it seems as if there's nothing we can do to slow down this process. However, that is not the case. The rate at which we lose our epigenetic information, or in other words, the rate at which we age, is largely determined by our lifestyle and environmental factors. This is why running every day, cutting out sugar from your diet, and generally maintaining low levels of stress actually makes someone live longer. It just means the rate at which the epigenetic information is lost is slower, which on a cellular level means that their cells are more specialized. The in-depth explanation behind how dieting and exercise and meditation actually slow down and even reverse the aging process is incredibly complicated. If you're interested in learning more about that, I definitely recommend listening to the Lifespan podcast by Dr. David Sinclair. He really goes in depth in explaining aging and also how changing certain aspects of your lifestyle results in longevity. I hope you found this video useful and constructive. If you enjoyed, please do consider subscribing, and thank you very much for watching.